Great. Thank you to the Katarinas. Um, I'm really happy to introduce our next speaker. So Joseph, or JJ Richardson, received his bachelor's degree in philosophy and his master's in industrial and systems engineering from the University of Florida. JJ then completed his PhD in chemical and biomolecular engineering under Professor Frank Caruso at the University of Melbourne in 2015. Since then, he has worked on both industry and academic projects at CSIRO, the University of Melbourne, and the University of Tokyo. He's currently a JSPS Fellow under the supervision of Professor Ijima at the University of Tokyo, and his current research focuses on metal-organic materials for biotechnology applications. And with that, I will turn it over to JJ.
All right, well, thank you for that. Yeah, I've made this talk very conversational to match the theme of this being a conversation. So if anybody wants hardcore scientific details, you can email me or you can ask afterwards. This is one of my nanobionic experiments. It's a wilting flower. It sort of like maybe symbolizes the process of science. Great hope, some beautiful fluorescence there, and eventual decay as we move on to other things. So I want to start with porous materials because nanobionics are all about essentially porous materials and interfaces, because that's how we can control our environment. So porous materials are incredibly important in nature. They give hybrid properties, anything from capturing moisture to capturing gases to increasing robustness. For example, with sandstone, eggshells are a beautiful example of having pores, but being incredibly robust in many dimensions, yet weak enough for a small animal to crack out of it. Butterfly wings get structures from their pores, and synthetic porous materials, what humanity has tried to adopt these things from nature, has led us to fantastic innovations that essentially impact our everyday life. So things from insulation to essentially bricks, and if you'll see on the bottom, cakes and breads, which I view as very much synthetic materials. But if you've ever had a cake that's not porous or bread that's not porous, essentially it's more akin to the brick up there in the top corner than it is to an edible food. So those pores are really important for, locking in moisture, keeping gases there, sequestering colors, thermal insulation, holding soap for sponges, and essentially they allow for the transport of materials across some interface. So the porous materials I want to talk about today in this conversation are often termed MOFs. It's not IUPAC defined, it's not a great definition, but essentially it's a metal organic framework. So you have a organic linker. It can be very, very small. It can be quite big and multidentate, and some sort of inorganic node, whether it's a metal ion, whether it's a cluster, whether there's water around it. There's a lot of flexibility in what you construct these from. Essentially, you can choose any metal from the periodic table, and then each metal will have a list of corresponding organic ligands. There's over, I think, 100,000 identified to date. Not all of those made physically in the real world, but there's a lot of potential. So that's what I use for getting pores from my nanobionic materials. MOFs have tons and tons of applications, which is why I've identified them as a great way to create these nanobionic hybrids. They, as you can see there, it's a list. It's like what every super material can do. It can do anything, but notice that the applications are in quotes because very few true applications have been realized in industry. There's about 3 products on the market utilizing these MOFs, but there's probably a few million papers. There's, 10 or 20 science papers every year using these. So intellectually, they're very interesting, but we've had trouble really transitioning them into the real world. Now, these MOFs, often the reason why they haven't transitioned into the real world is they can be very fiddly to synthesize. Often it requires solvents, high pressure, high temperature, because often they're also crystalline. So it's like growing crystals in nature, but we just have to do it in an accelerated fashion in a lab. However, colleagues and myself noticed that if you have seeding agents, you can grow these metal organic frameworks very, very rapidly in aqueous conditions. And once we're in aqueous conditions, we can start integrating them with life, right? That's the crucial media for all living things is water. So whether you have synthetic polymers or biomolecules, they can sort of act as a sink for the metal ions, which concentrates them locally, so those organic ligands can start bridging them into a crystal. So having that sink really helps. And we extended that to living entities because they're very rich in biomolecules. And although their radius of curvature is much, much, much larger than a biomolecule, these metal organic framework materials can still seed on them. So that's the porous material. That's the material we want to create our nano-bionic hybrid with. But what do we use it for? So this is just a very simple summary of how important interfaces are. So porous materials are crucial, but so are interfaces. If we remove our interface, we can't interact with the world, whether that's our skin, whether that's our cornea, whether that's the cup I have my water in. There's a lot of interfaces holding the water in there rather than having just pour through the material all over my lap. Your phone may have a touch screen. That interface is really, really important for connecting you with everything that's in your phone. So coatings let us engineer interfaces. They let us engineer the way we interact with the world. From very simply frosting on the cake you might have, which keeps that moisture in those pores, to a contact lens. If anyone has that, there's coatings to keep that wet so it doesn't dry out your eye, so it doesn't scratch up your eye, so that it can heal, to resins that seal wood so that our buildings can last a lot longer, to coatings and nano coatings that we put on our body quite regularly, whether it's cosmetics or sunscreen. So engineering these interfaces allows us to engineer the way we interact with the world. Now, for nanobionics, I haven't defined those yet. They're essentially any nanomaterial that's integrated with a living thing. So the most sophisticated, probably science still in my view, that exists is the cochlear implant. It's a true bionic construct. My dad has two, one for each ear, and it really is like the epitome of hybridizing what it is to be human with what it is to engineer things as a human race. So I want to take you through the process we go through when we're creating one of these nanobionic constructs. Clearly not with humans, that's incredibly, incredibly challenging. But in this, I'll be talking about plants. So multicellular organisms. I'll show some examples of unicellular organisms at the end. It's not as scientifically challenging to do with unicellular organisms. So if we break down a tree into its constituent parts, we see that it's made of wood, which is made of essentially small cells. Cell walls of those might have cellulose fibrils, then cellulose nanocrystals, which compose those, might be cellulose sheets, all the way down to cellulose chains and a few small repeating units of sugars. So I'm going to take you on this journey, how we make nanomaterials at the interface of all of these, and how we check their porosity and effectiveness for various applications. So starting with cellulose, we found that cellulose and other polysaccharides can act as a sink to concentrate metal organic frameworks. So these metal organic frameworks we use were just 2-methylimidazole and zinc. Super easy. You mix them in water at essentially isotonic conditions, and rapidly you start forming crystals. Interestingly, the crystals can have two different isoforms. I won't have too much jargon in there, but that ZIF8 and the ZIF-L, it's the same constituent building blocks, but they take on different crystal shape. Sort of like using food analogies, because it's very early in the morning, when you mix eggs and flour, you can turn them into pancakes, you can turn them into bread, you can turn them into cupcakes. It's the same building blocks, but you get different forms out of it. So we can use different polysaccharides, which will also control the structure, but we use dextrin here, we use pallulin, and As you saw, we just vary the concentration as we move right. It's not too important the exact conditions, but the crystal size and the crystal form change as we vary the precursor concentrations. That's important because some of these isoforms are porous, which will allow fluid transport through the crystal so that our living entity can still interact with its environment. And some aren't, which means it's essentially isolated from the environment. Not that that's always a bad thing, because like we saw with egg shells or sporulation for certain organisms, being excluded from your environment can also be a survival mechanism that's very useful. So that was just a brief look at our study on very small polysaccharides. The cellulose nanocrystals are a bit interesting, and we can make these very beautiful large aerogels from cellulose nanocrystals in moths. And the way we do that is essentially we extruded cellulose nanocrystals just with a pipette or a syringe or whatever you want into the precursors for the moths. And like I said, they often are very hard to form these moths without some seeding molecule, without a sink. And so no crystals form if you just have those two ingredients. But once you put in those cellulose nanocrystals, immediately you get crystallization along that interface, which allows you to make these beautiful hydrogels. This is some pictures of those fibers. That really strong white is coming from that metal organic framework. It scatters a lot of light, so it looks very, very bright white like milk. And if we zoom in on those fibers, we can see that we have these crystalline particles there. What's even cooler is we can actually see the individual cellulose nanocrystals, and sometimes when they assemble into fibrils, interacting with the moth. And the moth, if we freeze this in time, we can actually see that these moth crystals are growing truly around those cellulose nanocrystals, and that it's almost like fingers that the moth is growing around. So this showed us that we truly are creating this on the interface of this material. These interfacial reactions are what was really important. We did some catalytic applications with these, with the fluid transport, but I won't go into that because the more exciting things is as we get up towards the organism. Cellulose fibrils, so we just use bamboo, so increasing in complexity here. We form these, and as you can see, they're not really crystalline at this stage because the radius of curvature now is in a very awkward stage for these crystals to form. So they're amorphous materials at this point, and these fibrils essentially cause the particles, these seeds, to agglomerate and grow along the fibril. As you can also see from that, it's not fully contained. So the fibril it's sort of like a Janus particle or binary, where you have this fibril and then you have the particles all on one side of that. Some fibrils, if you look in, are completely coated, some aren't. And a lot of this has to do with wetting phenomena and adding a little bit of solvent to make sure those precursors mix better. But it shows us that there's a lot of promise for scaling this up to more complex, larger systems. So moving up the complexity chain, we're finally to cell walls. This is where it starts to get interesting from the terms of nano-bionics. We're not just making a material anymore. We're making truly a living material hybrid with a nanomaterial. So this is a little bit more scientifically complex, but there's only 4 pictures, so hopefully we can all get it. But on the top, we have the moths are labeled in this bright blue. And on the bottom, we have chloroplasts, which can glow red under a microscope. And if we grow our moths around these cell walls, which is the left-hand column, we can see that we have, beautiful moths all around it. Of course, if we don't grow the moths, if we just put one of the precursors in, for example, just zinc or just 2-methylimidazole, we don't see any of the fluorescence that we would get from this moth. We see that the coating around in that C panel is quite strong. It looks fairly uniform. There's some blotchiness, but for Z sections, it turns out that's more of a focal issue. And if you look really hard, you can see that actually the chloroplasts are coated too. We can see these from electron microscopy images. So this was our first evidence that we truly could coat more complex living things. So trees, this is an actual picture of a tree that we coated. No, I'm just joking. That's clearly we're not going to coat something that big. It would be probably terrible for the environment too. But once we get to trees or complex plants, it's a lot harder to just dip them in whatever you want and have them be a seed. So it was the first time we had to think about actually synthetically, how do we want to incorporate our precursors with our living object? Because now the living object is a little too big to put in unless you have a giant bathtub, which at that point, it's also a waste of materials. So we just used this, very ancient technique of dyeing flowers. So thousands and thousands of years old, we just essentially clip a flower and different capillary forces will bring a fluid up through the flower, throughout its stem, into the leaves, and into the actual flowering tissue. So we probably all did this in like primary school or something, but it's actually a very useful method. This was inspired from a very cool paper that our group did not do about integrating a conductive polymer with plant tissue. And it was really fascinating. It was in Science Advances, so it got decent publicity, but it was sort of that look that, oh, we can take these really complex organisms and we can put nanomaterials and get some unique property that's outside the realms of biology, like conduction. This is something that evolution really hasn't produced, but synthetically, humanity makes conductive materials all the time. It's something we're very, very good at, but biology is not necessarily as good. So you can see here, it's quite complicated, but what essentially they use vacuum pressure, and they also use capillary forces to integrate this PSS, this conductive polymer, into the plant leaves. You can see the plant leaf looks a little bit nasty on the right-hand side, but still very cool experimental concept. So we took something very similar, and first we tried plant clippings because that was easy. We knew that protocol from childhood that we could get materials up into the stem, and we incubated them with two different metal organic framework materials. One we did in one step, which was that zinc and 2-methylnitazole. The other one we used rare earth metals, so that LN for lanthanide metals, and this benzyl dicarboxylate, because that has some inherent fluorescent properties when they're coupled. And for the one step, the xylem essentially would just suck these up, and as the ingredients went up the xylem, they would start seeding and crystallizing all throughout that. It wouldn't seed in the reservoir of fluid, it would only seed once it came into contact with the biomolecules. The two-step forms much, much, much quicker, which is why we had to go to 2 steps. And we tested infiltrating the metal first, or infiltrating the ligand first, and did some studies that I'll show you on another page. But what we saw essentially was that as you went up the flower and as you spent more time, you got bigger crystals, and you got these different crystal forms. This is just showing you that ZIF8 and ZIF-L again, but more in a more chemical sense. You can see the periodicity there. So this is my favorite picture from all of them. This is essentially SEM, it's a cross section, and we have the elemental mapping there. The zinc is only from the moth, and the nitrogen, of course, and the oxygen and the carbon are mixed. The oxygen is just from the plant, but the carbon and the nitrogen come from both the moth and the plant itself. If we look at the false color, that's where you really see all these crystals everywhere. And you see them on the outside of the plant, which is that top part, and then you see it in the xylem, not as much in the phloem, which really shows that this is happening as the materials being taken up. If we compare that to a naked plant, you can see the naked plant is very, very smooth. The outside is very, very smooth. The inside is very smooth. We don't see these crystalline particles. So we did some more elemental analysis, and on the left-hand side, these are the plants that have the moths in them. You can see that the nitrogen and zinc composition is incredibly high compared to native plants, above sometimes, 10, 20%. This is interesting in terms of showing how much of the material is actually moth, but also, of course, nitrogen can be very useful for plant growth. So that's interesting also. This is just a big data dump from the synchrotron to show you that we did do more hardcore science. Since this is very conversational, I want to give it some scientific legitimacy. But we used the Australian synchrotron to look at the scattering profiles of different plants, roses, reeds. We also did some grass as you go up the plant. So starting, very top to very bottom, taking different time points. And we actually saw different crystal isoforms appear at different lengths and different distances and different times. So it showed that we essentially, when we get the moth to seed, we get one form, and then as this incubates, it transitions into another one. So it was quite interesting. We also saw different isoforms depending on if it was woody tissue or not as woody of the plant. But yeah, lots of crystals in the plants. So if we use that two-step process I mentioned initially, we can get these fluorescent plants. So again, something that's not too typical in nature is fluorescence in plants, but we can see that if we put the terbium in this case first, we actually didn't form any moths, and that was because the terbium moved much quicker through these capillary forces than the large organic ligand, that benzyl dicarboxylate, or BDC. But if we put the BDC first, it infiltrated that for about 12 hours and then switched it over to a terbium reservoir, we could get these nice platelet fluorescent moths growing throughout the inside and outside of plants. Here, this is like a water lily pretty much that we did. And you can see from the inside, it's incredibly bright, even in daylight, if you have a fluorescent lamp. It's very bright. And by changing the metal in G, we can change the fluorescence from green to red to orange. So this is a little bit of our application, because as you scientists know, every paper nowadays needs some application, no matter how fake or real the application is. But what we wanted to do here was show that we could make plants that essentially could sense acetone, so sense some organic solvent that was very toxic to them and change their fluorescence profile. Later, my colleague who I did this with, he's done like a smartphone app that can sense this and all these things. But realistically, this isn't useful for crops or anything, because the cost of the ingredients that we're using in this instance are more expensive than the crops you would get from it. But as a proof of principle, in dissecting the leaf, we could see that if we cut the leaf in half and put half in acetone and half not, our fluorescence profile would vary by about, we get a 50% drop in the fluorescence upon incubation with acetone. And we could sense down to, I think it was about 0.2% acetone in water using these. Lots of cross-sectioning, lots of UV lights. Cameras don't really capture that well, especially I was often using my smartphone, which, just so you know, will burn out the chip on it, so your selfies will look a lot worse afterwards. But we saw that we could truly, with this, integrate functional material with a functional organism, a plant. I didn't show here, yep, but this was all destructive, where we were cutting a plant, we were essentially killing it to do our experiments. But we also did experiments with the roots intact, where we essentially picked out a weed, washed all the soil off, and then incubated it in our two-step process. And we could also get the fluorescence throughout the plant and keep the plant truly alive, where it could propagate. So the last slide I want to show is the unicellular organisms, just to show that there's a lot to do there. There's a lot of almost cooler stuff you can do with single-celled organisms. So heaps and heaps of papers on this over the last decade that I've been involved with, everything from making magnetic cells in the bottom right to making cells that have a proteinaceous coating around them that can allow them to target new things. As you see in the very top, we have these sunflower seeds, almost looking things. Those are all yeast with different nanocoatings on them. The nanocoatings can protect them from organic solvents, from fungicidal agents, from enzymes. And with a lot of those, we've made degradable shells. You just add a chelating agent and all of a sudden your yeast are free and can propagate again. We also, again, push the confines of biology to put an enzyme in their shell that would convert something they couldn't live off to something they could live off. And then we had almost like space suits where they were in a very harsh environment, but they could make their own food using this nanobiotic casing. It's a different story though, different cool stuff. So it brings me to the end. The conclusions of this talk is there aren't any. This process is still going on. We're not done thinking. We haven't given up. That flower that was wilted at the beginning will blossom again. There's other cool things I'm doing here. It's still a little too early on. And of course, there's a lot of incredible scientists that have been involved in different aspects of this work, and more that I can't thank. So if you're interested in this field, if this was just a conversation to sort of whet your appetite, please feel free to email me. There aren't too many groups in the world working on this, but it's a space that's really ripe for interesting experiments. So yeah, that's it. Thank you. Please feel free to ask questions.